Thank you for the invitation to present the GENOMIT approach. And actually, this is the main picture about the GENOMIT consortium and the GENOMIT approach. So it's we realized already 15 years ago, or more than 15 years ago, that when we are dealing with rare disorders, with mitochondrial diseases, that also we had established national networks, that one country is too small to study rare mitochondrial diseases. We have a number of mitochondrial disorders where we see just very few patients per country, and if we want to understand more about these diseases, we need to collaborate beyond the national borders. So, we started with 5 countries, putting our resources together, and now in the moment we have 23 partners from Europe, Canada, USA, Japan, and China, and we started in harmonizing our national registries and building a common registry in which we can feed in all the data we had collected already before, and also in a prospective manner where we can enter the clinical characteristics of the patients which are seen at the different centers. So if we count all the patients which are in the national registry or in the GENOMIT registry, which are kind of shared by the GENOMIT consortium, we end up with 15,000 patients, which is a number which now really allows to better understand single disorders where before we had very limited resources. And here you can see on the pictures the main actors of the GENOMIT network from the current and last funding period. So this large number of patients in the registries allows now much easier and faster than before to study the natural history of diseases, and this is just some examples. So it's not that we always have all the information we would like to have for natural history in the registry, but it is a big step to start the natural history studies, because we can easily see how many patients we have with a certain diagnosis, and we get some core data about the phenotypes. There are often specific phenotypes which are not in the registry, so we have to go back to the clinicians to get more information about these patients. But this is enhanced natural history studies in Europe and give us the power and the numbers to do these studies. One important aspect is from the beginning on, we were closely collaborating with the national patient organizations and then with IMP. And they are, so we have about 2 meetings a year, and always the patient associations are invited and join us, and we have a very nice collaboration together. So I put some interesting, what I found interesting points here when it comes about the registry. So we have 1 variant, nearly 700 patients, 1 gene, nearly 900 patients, or 1 disorder, more than 1,000 patients. And I will go to some findings also from the genetic point of view. So MELAS is the example with 1 variant, and this is the study coordinated by Costanza Lamperti, looking into the natural history of the MELAS patients. And you can see here the contributions of the different partners from the GENOMIT network from the different countries. And this data which are there collected allowed Costanza to look what are the phenotypes of the patients which predict stroke-like episodes. And this is work under progress or under investigation, but it helps a lot to better understand the progression of the disease and what are the phenotypes which indicate these strokes. Another example is, and this is the most frequent nuclear gene which is affected by mutation, is the PDHA1. And we just published last year a cohort of nearly 900 patients with mutations in this one gene, and this was retrospective from the literature collected cases and newly collected cases, and it's about half of the patients which have been put together here, and it started also with a registry which we have, but then extended also to the friends and colleagues outside of the GENOMIT registry. Another example is Lee syndrome. There is the Lee syndrome as disorder, and we have it was presented already several times, so I don't need to go to the phenotype. And here we have more than information for more than 1,000 patients, and the cause are more than 100 genes. So this is the distribution of the patients from which we have the information and the numbers. So we have the information for more than 1,000 patients, and we have detailed information from 400 patients where we really have for each symptom the age of onset, that we can better study also the progression of the disease. And these are the initial symptoms and how they distribute over the cohort of these patients, just as an example. But we can also much better look now in the survival of the patients, how much it is depending on the gene defect. And I just remember that we spoke about a clinical trial about taking patients with ATP6 mutations. And you can see, I don't know if you can see here for ATP6, there are three different variants. And you can see that there is, if you look on the survival, it makes a big difference if you include patients which are very young, so several months, or if they are 2 years old, because in the first 2 years, most of the patients which die will die. And this has an impact on the design of the study and how to interpret the data. And with this data, we can also model or look into the factors which have an impact of survival, like the MRI pictures, the gene defect, the age of onset, and what are the initial symptoms, to predict the survival for 3 or 5 years in a quite good accuracy in this data set. So again, something important for any clinical trial for patients with Lee syndrome. So these are the data we have from the clinical point of view, but in this GENOMIT consortium, and this was one picture how we, from an application in 2022, where we and this is the second part which I want to pronounce here, is that we not only have clinical information, but we have put together more than 3,000 exomes from patients with mitochondrial disorders, which in the past resulted already in the discovery of more than 60 novel disease genes in this cohort of patients we share. But we have also genome sequencing data. We have genotyped now more than 3,000 patients with mitochondrial disorders. We did RNA sequencing from fibroblast cell lines and proteomics. It's now we have data set from more than 600 fibroblast cell lines where we have these multi-omics, and we can increase the diagnostic yield by more than 20% by doing RNA sequencing and proteomics of the unresolved patients after exome sequencing. And we have done metabolomics and lipidomics from about 3,000 patients. These are resources which are available to study, to search for biomarker or other important factors, modifying factors, factors affecting penetrance which goes beyond the monogenic diagnosis. And so this is a network, and the network is not only from clinicians, but every center has a specific focus. So we are very complementary. As you can see here, the different expertise from the different centers, and the regular meetings of this group of GENOMIT partners allows us to discuss the projects early on when we start the project, to exchange ideas, to comment on strategies, and to be much better focused. And there is an atmosphere also of trust and sharing, and this brings us a lot of advantages. I just wanted to mention as an example, we had this variable study in MITO, and now we extend this in the GENOMIT consortium because we think they are promising data to have a better continuous natural history study. And this is, we started with variables, and now we use a mobile app, and this is actually some former students founded a company which is now involved in these studies. Coming to the genetics, this is a very every genetic lab is going this kind of procedure. So there is a phenotyping of the patients, then you do exome or genome sequencing, and then there is the American College of Medical Genetics criteria, how you analyze the genetic data to come to a likely pathogenic or pathogenic mutation in the patients. And in this, when you go follow this street and criteria which are defined, we can solve about 55% of those cases which have not been solved before by exome sequencing, but we increase this by another 20% with multi-omics, but we still have a large cohort, about more than 1,000 patients, where we do not have a diagnosis. So this was a picture from a few years ago. We do find in the majority of patients a cause in a mitochondrial disease gene, but there is also 15% of the suspected mitochondrial disease patients where we find genes not known to be associated with mitochondrial diseases. And there is a fraction of about a third where we have a candidate gene where we need to extend the studies within the consortium by functional studies, by identifying more patients, to have the evidence that this is a novel disease gene or a novel disease variant. But not always we have a candidate. Within the consortium, there is also always some new discoveries, and one which is, let's say, new in the last years, that so far we was always focused on protein coding genes, but there is a number of RNA coding genes which play a role in splicing. And these very general processes like splicing, if there is a splice defect, they can also affect the mitochondrial protein. And in this case, we saw a defect in the splicing in a complex 1 subunit. So it's an indirect complex 1 deficiency and mitochondrial disease. We also, when we put the large data set together, we gain the power to find digenic inheritance. So it's the chance that two genetic variants in different genes come together is statistically much more rare than a monogenic disease. So we need really large cohorts of patients to identify digenic or digenomic mitochondrial DNA mutations in combination with nuclear genes to explain the phenotype of the patient. But with this large data we have, we can find these kind of observations. Also, the collaboration was key to get this amount of fibroblast cell lines where we did omics, and it helped us to develop tools to analyze these omics data. And there is, on the bottom, there is a number of publications where we improved the analytical tools and AI tools to analyze transcriptomics data to find the disease-causing variants. And the same is also having the fibroblast cell lines. We not only did transcriptomics, but also proteomics, and also there we are publishing tools how to integrate these different omics layers and to identify disease-causing variants. So besides providing diagnosis for patients, we also improve the technology for any kind of diseases. Something which is a new, or it's the non-geneticists may not understand this picture in the first view, but it's a very famous picture in genetics. So we have here you see on the x-axis you see the frequency of variants, and our understanding is that only a rare variant which has a frequency in 1 in 1,000 or even less can have a strong effect and be responsible for monogenic diseases, while these variants which are quite frequent, sorry I can't hear, these variants which are quite frequent, they have an impact on common phenotypes or common disorders like diabetes, or Alzheimer, Parkinson. So, so we are searching in the space of rare genetic variants, and this is also defined by the ACMG criteria that we are looking for rare variants. But not everything can be explained by the monogenic concept which are defined by the ACMG criteria, but we see variants with reduced penetrance. And variants with reduced penetrance have a higher allele frequency, and usually they are filtered out, and we miss them in our consideration. And so this is an area where we now also find pathogenic variants which are quite frequent, but they have a reduced penetrance or a special constellation of alleles which then cause the disease, but they are difficult to find, and we need large numbers to have a statistical evidence to convince the pathogenicity. And this is in the space of unsolved cases, we can find this kind of combinations of variants. Another example for an indirect effect is MORG2. It was one of our most frequent findings after exome sequencing, but we couldn't link MORG2 to mitochondrial disease because it's a factor involved in the epigenetic modification of the nuclear genome, and there was no hint for an interaction with a mitochondrial dysfunction. So there is the phenotypic spectrum of MORG2 patients is that about a third present with CMT, another third with mitochondrial or Lee syndrome, mitochondrial defect and Lee syndrome, and another third with cocaine syndrome, SMA, or other phenotypes. So we asked the question, and we could ask this question because we had an increasing number of MORG2 cases. How can it be linked to mitochondrial dysfunction? And what we found is that in these de novo gain of function mutations, there is a specific increase in methylation in the promoter region of a gene which codes a complex 1 subunit. So the epigenetic changes in the genome switched off a gene which is needed to assemble complex 1, and the patients have a complex 1 deficiency which explains the presentation of Lee syndrome in these patients. So it's again an indirect effect on a gene which is not related to a primary mitochondrial function that can impact energy metabolism, and in this case it's the epigenetic modification. And what is not possible from the genetic point of view, so you can see here on top there are 2 variants and they have been associated with CMT or with Lee syndrome or with SMA or cocaine syndrome, and the mutation itself doesn't tell us what will be the phenotype, but if we consider the methylation together with the mutation, we can predict what will be the phenotype. And the increased methylation causes then the Lee syndrome. Another example which we can do in these large cohorts which we have is we collect plasma samples in a regular way. So we have more than 7,000 plasma samples collected in the last years, and we selected 3,000 of them to do metabolomics and lipidomics, and we analyzed these in an untargeted fashion. So we can ask, are there biomarkers for mitochondrial diseases? And we could identify molecules which have a high predictive value of different mitochondrial diseases. We had to establish this assay because it's a metabolomics or lipidomics assay in different platforms, and we was able to do this now in at least 3 different platforms in the Netherlands, in the US, in Germany, and now we was able to identify what is the molecule, and now we are developing a specific assay that we do not need to do a metabolomics or lipidomics experiment. So these were some examples what you can do in this international collaboration, where you have numbers to have power also to have a statistical analysis, and this is done by this nice group of people where we are always happy when we have a meeting to see each other, like today here. And for this I would like to thank you, the Mitocon organization, to invite us. It's very important that we have this exchange of ideas and concepts and even material and data. So thank you very much to Mitocon and all partners.